we may. Okay, we start to discuss about our topic today. As I explained before, that we will focus to discuss to answer these 5 questions in our class, and I think we will randomly ask the volunteers to answer such a questions. So you may try to suggest an answer or explain or comment based on your understanding on such materials. So it is really the common concepts that muscles is the major factors as a major task force to do any kind of talks directed by muscles by nervous systems. So there is the neuron, the particular neurons, call it motoric neurons, that function to stimulate, to direct, to sense a message to muscles in order to generate such an activity. So when we talk about the muscle, it is really simple. Actually, the activity of muscles only two types of the muscles activity: contractions, and afterward follow it by the relaxations, and then contractions again and relaxations. So by these mechanisms, we then could move, we then could also lift something, and we could walk, and also we could speak, because the muscles in our vocal cord and also the muscles of our tongues also involved in such a contraction and relaxation. So therefore, many life process in our body and also in animals involving such a muscles activity. So our first question is, since that nervous system has a close relationship with muscles, so anybody could explain about how the mechanisms that nervous systems could control and could send information to the muscles? Mifta, please, you have any suggestion for such mechanisms? Please.
Okay, sir.
Okay,
okay,
thank
sir.
you.
Can you hear my voice, sir? Because my connection internet is not stable.
Okay, that's okay.
Okay, thank you, sir. I wanna try to explain how the mechanism of nerve system to make contraction or relaxation for the muscle.
Mm-hmm.
So actually, it is have a relationship with it is similar. It is similar with
Mm-hmm.
the how mechanism in nerve system between neurotransmitter and in synapse.
Yeah.
But the differentiation of the nerve system last week we have explained that the presynapse in
Nervous
in
systems.
this
Mm-hmm.
no, in muscle system, sir.
Okay.
In muscle system, that neuron motor neuron axon, and then the postsynapse in here muscle. So that's the different the simple one to make it different. So how to make what the mechanism of this one work?
Mm-hmm.
So motor neuron axon that have so many calcium in there, it is have connection and relationship with the axon potential. So in the presynapse, I mean in here motor neuron axon, in there is so many vesicle. In vesicle there is neurotransmitter, but in here we can call neurotransmitter with acetylcholine. So when calcium that have more intensity in the motor neuron axon, so that how to say memicu this acetylcholine to go to outside. So because of that, acetylcholine go outside, and then that's make so as we know, each neurotransmitter, or in here acetylcholine that have like a receptor. So
Mm-hmm.
in here, the receptor of acetylcholine we call with nicotinic acetylcholine receptor.
Mm-hmm.
So this when acetylcholine meet with the receptor, so it's make postsynapse in here muscle melengkung. How to say it? Sorry, make the postsynapse melengkung. Because of that, acetylcholine menempel with the DAM receptor, and that memicu terlepasnya kalsium karena adanya potensial aksi, yang mana kalsium itu bisa terlepas dari setidaknya ada dua tempat, that's extracellular calcium and also in sarcoplasmic reticulum. When calcium outside, so calcium that will make calcium will menempel with troponin, and that memicu kepalanya myosin itu untuk menempel dengan aktin, untayan garis yang halus. Ketika kepala myosin itu menempel dengan untayan yang halus, maka disitulah terjadinya contraction mechanism. So, when calcium not go to outside from extracellular or from sarcoplasmic reticulum, so just still inside, so that will not tidak akan memicu contraction mechanism. That means it is relaxation mechanism. I think that's one, sir. Thank you, sir.
Okay, thank you, Mifta. So based on your explanations that yeah, it is correct that actually such a functions is associated with the structures, the particular structures that's called neuromuscular junctions. So when we try to focus on these structures, like you may see in the screen right now, that between the neurons, the motor neurons with the muscles, this is muscle fiber, there is a relationship that's called synapse. So this is chemical synapse. Synapse mediated by the chemical substance called neurotransmitter, and for the these neurons and muscles, the particular neurotransmitter is acetylcholine. So here it is when we take a look in particular part of this. So this relationship, call it the neuromuscular junctions, composed of the terminal button of axon promotoric neurons and the particular part of the membranes for the muscle fiber, that's called motor implant. So can you see here? So you see here that actually there's a particular structures, textures of the surface of such membranes in motor and plate. Here is the regular surface for the muscle fiber. So it is like a smooth, smooth surface. But for the muscles and plate, the surface is like, there is a kind of crease. There are several creeps here. So it functions to increase the surface area, because this is a kind of the specific locations for this activations of several ion channels. So as previously explained by Mifta, that actually it is involved in neurotransmitters, and when neurotransmitters is released through the particular inductions from the electric stimulation from here, it will induce the binding of neurotransmitters with its receptors associated with the channels, the ion channels. So this is sodium channels, and therefore it will induce the increase of membrane potentials in muscle fibers, and therefore triggers the electricity transmissions from from neurons to the muscles. So basically, the muscles is activated by the electric, and therefore when you are stunned by electricity, for example, when you touch the current from the electric, it will induce a kind of stunting for you. So because your muscles directly charged by such a electricity, but in this position, in this process, particular mechanism happen in our body, it is related with that the electricity coming from the neurons, and then it is transmitted by neurotransmitters, and therefore it will generate the electricity in muscle itself. So this is something unique about the muscle, because the muscles is quite similar with the neurons, because muscles could be activated by the electric, and muscles also composed of these ion channels with a particular sodium ion channels and also calcium channels, and with larger composition as compared with other types of the cells. So therefore muscles could also work based on the electricity triggered by the neurons. So that is, I think, the correct answer from Ifla. Of course, it is this relationship is mediated by neuromuscular junctions, and therefore the neurons could induce the activity of neurons. I mean, muscles based on the stimulus, based on this impulse or action potentials that will travels across this synapse or neuromuscular junctions. And Aryan, do you have any additional explanations? Okay, so next we will move to the next questions. Let's move to the next questions. We will discuss about, sorry, we need to open the correct slide. So okay, so here it is. We will move to the next questions. So, okay. So when we talk about the activity of muscles, of course, it is only composed of. Okay, Ravia, do you have any questions, or oh, you want to answer such a question, right? So the second question is that whether what are the requirements for the activity of the muscles? So whether, or what kind of the particular needs and requirements in order to induce the muscle contractions and relaxations? So Ravia or Aryan will be the first. Anybody? Okay, Ravia, please.
Okay, sir. For the activation of the muscular is have the filamentin and the filamentic. Yang mana filament tipis itu ada berupa aktin dan filament tebal itu berupa myosin. Dan pada aktivitas otot itu adanya kegiatan antara aktin ataupun myosin, yang mana pada aktin dan myosin ini juga terdapat misalnya terdapat juga ada pita I dan juga pita A, yang mana pada pita I itu didominasi oleh aktin dan pita A itu filamen tebal yaitu myosin. Jika pada kegiatannya tersebut terdapat kegiatan kontraksi ataupun relaksasi, yang mana terdapat juga pengaruh-pengaruh dari ATP ataupun dari ADP. Di sini juga ada respon dari channel kalsium, yang mana jika yang seperti yang telah dijelaskan oleh Miftah tadi, neuron transmitter itu yang mana asetilkolin akan memicu terbukanya saluran sodium, yang akan sodium itu masuk dan memicu muatan positif di dalam, sehingga terjadinya potensial aksi atau depolarisasi pada otot. Sehingga tetapi kegiatan kontraksi maupun relaksasi ini tidak terjadi secara terus-menerusan karena adanya enzim asetilkolinesterase, yang mana itu akan menghilangkan transmitter agar tidak terjadi potensial aksi secara terus-menerus, Pak. Dan kalau pada kegiatan kalau pada mekanisme relaksasi serabut otot itu adanya perilisan dari ATP dan juga akan dimasukkan kembali ion kalsium yang telah keluar tadi sehingga tidak terjadinya kontraksi antara aktin dan miosin pada kepala miosin tersebut, Pak. Dan ada ion-ion yang terlibat dalam aktivitas otot itu ada juga magnesium dan juga troponin. Troponin ini merupakan bagian dari aktin yang mana itu tempat melekatnya kalsium, Pak.
Okay, thank you, Ravia. That's also based on Ravia's, answer. There is a structural requirement actually, so there should be actin and myosin filaments, and also there are other components needed. ATP as energy for, contractions, relaxations, and also ions, including calcium, sodium, and magnesium. So, Aryan, do you have additional or the similar answer for that? Probably you have another idea about the functions or requirement for the contractions and relaxation of the muscles.
Jawaban yang sama, sir.
Okay. Okay, have a similar answer, right? So, but when we take a look into the upstreams, so into the upstream process, so let's start from neuromuscular junctions. If we will talk about the contractions and relaxation, we should start from the very upper place, like the upstreams process. So in the upstream, it will require the inputs. So the bursts actually should be inputs or action potentials from the neurons. So action potential from neurons. This is the requirement. So without any impulse or action potentials from neurons, the muscles could then contract and relax. So it is also, understandable, for example, for someone having stroke that damage the brain associated with the motoric controls in the brains, and this person may suffer of paralysis. Sometimes it's at the left side or on the right side, both of hand and leg couldn't move. Why? Because there is no impulse, there is no exit potentials that could travels from the brain into the extremity, into the muscles of our legs and our hand, and therefore it is mean the requirement for the activity of such muscles couldn't be married with its needs, and therefore the first is impulse, and how such an impulse could travels from could be transmitted from motoric neurons into the muscle fibers. So it will need neurotransmitters. As Ravia stated before, that it will use acetylcholine. So neurotransmitter. So many toxins, many drugs also could block or could induce the activity, also could control the activity, contractions and relaxations of the neurons, of the muscles, by modulating this neurotransmitter. So several toxins from predators, they use the mechanism by blocking these toxins when it reaches the blood, and then eventually it will bind to the specific locations for the neurotransmitter acetylcholine, and therefore acetylcholine couldn't work properly, and after what there is no movement because the muscle failed to contract. And so it is also the next requirements. And so we start from the impulse and the neurotransmitters acetylcholine, and afterwards, of course, we will need the ions, the calcium, sodiums, and magnesium is additional. And afterward, of course, we will need energy for this, because the pumping process, the mobilizations of iron from outside to inside, or like mobilization of calcium from reticulum sarcoplasmic into the cytoplasm, requires much more ATP, and the attachment and detachment between actin and myosin also requires ATP, and therefore the ATP is needed. And so it is when we are, for example, under the starvations, or we just miss up our breakfast or miss our dinner, our lunch, and probably when we are fasting, we feel so tired that even we couldn't move properly and we couldn't walk in such a way much. Why? Because the requirements for the muscle contractions, for the muscle activity is lack. So the ATP is less, the energy resource is not adequate, and therefore we couldn't move. So there is no activity for such a muscle. So this is like when we take a look from the very upstream part in this sessions, in this particular. Okay, let's click here. So it will need a calcium ion, of course, and it will require the neurotransmitters, this impulse, and also the sodium ions, and of course the calcium in this one, and energy, also the ATP resource. Okay, thank you, Rabia and Aryan as well. And next we will move to the next question about so previously, as actually Rabia has explained as well, and also Mifa also previously has explained about the essential functions of the calciums. So here it is. We will move to the questions about what is the role of the calcium in contractions and relaxations of the neurons. Okay, Anissa, do you have any explanations about that?
Ya, Pak, terima kasih, Pak. Kalau untuk peran kalsium dalam mekanisme kontraksi relaksasi otot, dari yang saya dapatkan, kalau pada saat otot berkontraksi itu kalsium berperan dalam interaksi protein di dalam otot, yaitu pada aktin dan myosin. Dan kalsium ini mengatur kontraksi otot dengan proses juga yang ditengahi oleh yang namanya troponin dan tropomyosin. Yang dimana troponin ini tempat melekatnya kalsium. Yang kalsium itu melekat pada bagian yang membuat yang namanya binding site yang tertutupi oleh tropomyosin terbuka, yang dimana tropomyosin itu juga adalah protein yang menyerupai benang dengan fungsi menutupi binding site pada aktin ketika tidak berkontraksi. Jadi jika ion kalsium ini meningkat, itu dapat menyebabkan perubahan struktur troponin. Perubahan struktur troponin ini karena terikatnya ion kalsium akan menyebabkan terbukanya daerah aktif tropomyosin yang semula tertutup troponin tadi. Hal tersebut mengakibatkan myosin dapat berikatan dengan aktin dan membentuk aktomyosin sehingga terjadi kontraksi. Begitu juga sebaliknya pada proses relaksasinya. Dan dari yang saya tahu, bila kalau darah kalsium itu kurang dari normal, otot itu tidak bisa mengendur sesudah kontraksi dan tubuh akan kaku dan bisa juga terjadi kejang.
Okay.
Terima kasih, Pak.
Okay, thank you, Anissa. So based on Anissa, so let's take a look with the answers of Anissa. So Anissa, focus on this particular functions of calcium, because calcium expose the active site, the binding site for the calcium. Sorry, I need to enlarge this portion. Sorry. Okay, so probably we can. Okay, so take a look at these animations. So this is the head of myosin, and this gold ball is actin. So in contractions and relaxation mechanism, we notice that actually it is also manifestations of the binding between the myosin head with the actin. And when you see here, this is the yellow one is calciums, and this is troponin, and this is the yellow, the gold is actin. So the head of myosin should bind to this actins in order to induce the contractions and the relaxations afterwards. But during the normal stage, the basic stage, actually, this troponin cover. So can you see here that actually the troponin cover the binding site between myosin head with the actins, and therefore the calcium is needed in order to expose, to open such a binding site. And because the calcium, when the calcium bind to this site of troponin, it will induce the expose of binding site or active site for the actins, and therefore actin interacts in proper way in the correct positions with the myosin head, and it will generate the contractions. So can you see here that every time calcium bind to this troponin, and then troponin will change its positions, like to open the place of the binding site between the myosin head with the actin in a precise way. So it is one of the functions of the calciums in muscle contractions. So next, Anga, and then Fadila, we will also see. Anga, do you have other additional explanations about the functions of calciums in contractions and relaxations of the muscles?
Yes, sir. I will try to answer this question. Jadi, we know that the action potential in tubular triggers akan memicu pelepasan ion kalsium dari retikulum sarkoplasma ya. Dan increasing of concentration of calcium in ions in the cytoplasm of muscle cells will trigger muscle fiber contraction. Jadi sel otot akan memicu terjadinya kontraksi serabut otot. Dan untuk fungsi ion kalsium itu sendiri terdapat dua. The first benefit of ion calcium will attach to troponin in the chitin filament, which cause the active site of the actin to open. Jadi ion kalsium akan berlekatan pada troponin di filamen chitin yang menyebabkan sisi aktif pada aktin itu terbuka. Dan the benefit of second by ion calcium juga berguna untuk mengaktifkan enzim ATP yang akan menyebabkan ATP dapat melekat di kepala myosin, sehingga ATP dan fosfat dapat berlekat dengan kepala myosin. Dan kepala myosin akan melakukan pergerakan or mobilization or change so that muscle contraction occurs. Jadi pergerakan mobilisasi atau perubahan sehingga terjadinya kontraksi otot. Dan pada saat relaksasi ion kalsium is reabsorbed atau kalsium akan diabsorpsi beginning relaxation cycle or ATP is required. Jadi ATP dan fosfat pada kepala myosin itu akan tidak ada atau akan hilang, sehingga tidak akan terjadi interaksi antara kepala myosin dengan filamen aktin. Lalu konsentrasi kalsium di otot akan menurun, maka tidak ada kalsium yang dapat mengekspos sisi aktif dan dari aktin yang bisa berlekatan pada kepala myosin. Maybe not for me, and back to sir.
Okay, thank you. So Angga, similar with the previous answer from Anissa, right? So that you focus on that calciums, exposed, as I also have additional explained about it. That calcium is important to expose the correct positions, binding site between myosin head with the actins. And also you mentioned before that calcium is required to induce the activations of ATPase, so the activations of enzymes involving in hydrolysis of ATP to be ATP and phosphate. And okay, we can hear additional answer or explanation from Fadila, and afterward we will also hear from Alti. Fadila, do you have any suggestions about the functions of the calcium, please?
Yes, sir. I think that I have same answer with Angga and Nisa, but I will explain a little.
Okay.
from mechanism contraction and relaxation of the muscle, I think this is related with the concentration of the perubahan konsentrasi dari ion kalsium itu sendiri. That the what we call it, the munculnya the potential aksi dari manifestasi alkohol yang tadi yang dijelaskan oleh Mifta and Rafi also that make the voltage gate dari kalsium ini tuh terbuka atau channels itu open, that make the ion calcium mobilization into the sarcoplasm. Jadi dia dengan terbukanya gerbang voltage tadi itu membuat ion Ca+ itu bermobilisasi ke dalam sarcoplasm. That make the gradient concentration menurun. Dia membuat menurunnya gradient konsentrasi. And next, the ion after enter the ion calcium enter the sarcoplasm, dia akan berikatan dengan, we call it calmodulin or what CAM in the sarcoplasm. That make the phosphate terlepas dari CAM, and the complex of the ion calcium and the CAM that also make the mengaktifkan enzim myosin-like chain kinase or MLCK, that the enzyme will akan memfosforilasi gugus like chains atau rantai ringan dari kepala myosin, sehingga yang tadinya myosin this is inaktif akan menjadi aktif. Nah, selain itu juga MLCK ini juga akan mengaktifkan ATPase di myosin tersebut. Nah, dengan kondisi tersebut memungkinkan make the myosin untuk membentuk cross bridge dengan aktin yang we call it menjadi rangkaian dari kontraksi otot. Jadi so that's because the perubahan konsentrasi ion make the mechanism contraction and relaxation of the muscle. That's all from me, sir. Thank you.
Okay, thank you, Fadila. So there is additional explanations from you when we try to catch up what kind of information you add to this from the previous explanations, that the third functions of the calcium is actually to activate the enzymes that's a kind of myosin light chain kinase by combining with calmodulin protein in cytoplasms of muscles. So such a binding will you will see here in this process actually. So the calcium is really essentially needed, because when the concentrations of the calcium increase due to the enters of calcium from outside, and also calcium mobilizations of calcium from sarcoplasmic reticulum into the cytoplasms. So once this calcium ions increase in cytoplasms, it will bind with the calmodulins, and afterward, because the affinity of calcium with the calmodulin is higher as compared with the affinity of calmodulin with the phosphate, and therefore when the calcium meet with the calmodulin tend to bind with the calciums and will release the phosphate. So it's called the phosphorilizations. And this ligand, this binding, this complex between calciums and calmodulin is important to activate such an enzyme called myosin light chain kinase. So what is the functions of these enzymes? It will phosphorylate the head of myosins. So once this enzyme active, it will induce the utilizations of ATP, and then you see this from state of inactive of myosins or will be active states of the myosins, and the phosphorylated or active myosins and the active ATPases will triggers this kinds of these ones you see here, that the erections, so the activations of of the myosin head will change its conformations from like in sleeping state to be the wake up state, because there is the phosphorylations in head, and also the additional of ATPases activity. So in here we can see that the key role, the key functions of the calciums through this process, and therefore the deficiency of the calciums will impair these mechanisms, and eventually the myosin head couldn't be activated, and the ATPases also could not activate to hydrolysis the ITP, and as the consequence, as the results, the head of myosin will fail to bind to the actin, and therefore there's no contractions or relaxations in such muscles. Okay, Aldi, do you have additional explanations or similar explanations with previous friends, with your colleagues?
Yes, sir, I think it's similar with what my friends told before, but I think I have a question that it make me confused. Can I ask something, sir?
Of course, please.
About we know that channel is affected by maybe with ligand and voltage.
Sure.
But we know that when acetylcholine bind the receptor here, and it triggers some we know the excitation in the T tubulus. So the T tubulus, how can they the action potential travels along the T tubulus? So there is a channel here that make the excitation or the potential action flow to the SR, or it is just one kind of tissue, but it is consist by several channels here. I think this is my question, sir. Thank you, sir.
Okay, such a nice question, Aldi. Thank you for raising such a question. So as we see that actually this is sodium channels. So the surface of these membranes for the muscle fibers composed of these sodium channels, and these sodium channels will be open due to the increase of ligand-gated channels. So the first in these regions, the sodium channels is open because the ligand-gated channels, because the triggers from the binding between the ligand called neurotransmitters acetylcholine with the receptors, and therefore it will open the sodium channels. But after the generations of the potential action potentials, and then whether this surface is composed of another channels, commonly it is also composed of sodium channels. But the type of sodium channels here is not ligand-gated channels, but it is voltage-dependent gated channels. It is main these channels, the sodium channels through these T tubules, is composed of a kind of sodium channels activated by the voltage. So when the voltage arrives, it will open the sodium channels, and therefore the sodium will enter the gate, and then will influx into the cytoplasms of these muscles, and therefore the cytoplasm of muscles also will undergo a kind of increase of action potentials. So when you ask about the channels, it is commonly composed of sodium channels, but this sodium channels is opened by the increase of voltage. So it's so called that voltage dependent sodium channels. So the channels that only can be opened by the increment voltage. But in these locations, the similar sodium channels, but the sodium channels is open only by the activations of ligand. So ligand gated sodium channels. That's the channels that only can be opened by the binding of the ligand with its receptors in such locations. So here it is, as the consequence of such the increment of voltage, it will also the triggers of opening the voltage dependent calcium channels in these locations in the membrane, surface of the membrane, and also in this membranes of sarcoplasmic reticulum. So the membranes of the muscles and the membrane of organelles called sarcoplasmic reticulums, or in ordinary cells, it's called reticulum endoplasmic reticulum. It is also composed of ion channels. This is also kind of voltage-dependent calcium channels. The calcium channels only can be opened by the increment of voltage or action potentials. So that's I take the answer for your questions, Aldi.
Yes, sir, I got it. Thank you, sir.
Okay, thank you very much. And okay, so next we will discuss about the third question for this. Probably we'll move for the last one, right? Oh no, there's still two questions. We can understand by let's focus on how the muscle contracts and relax in skeletal muscles. So when we see here that actually when the impulse arrive into these locations, and it will triggers the polarizations of the muscles membranes, and of course it will trigger the electricity, or the alterations, the increment of electricity in the muscles of the neurons, muscles of the fibers of the muscles. And then it's called the increment of membrane potentials in the muscles fibers. And such a electricity, such a depolarizations followed by the repolarizations of these muscle fibers will afterwards triggers the physical activity of the muscles. So they call it twitch. So twitch is composed of one contractions followed by one relaxation. So this is kind of the regular cycles. So can you see here that after stimulus, then stimulus from the neurons, of course, the impulse arrives and it will triggers the depolarizations here, will triggers the electricity, the electric activity. That after the electric activity, it will follow it by the physical activity of the neurons, called it contractions and relaxations. So here is when you see the particular pattern in skeletal muscles that also we can find in our extremity regions. So for example, here in single stimulations from neurons, and then it will induce the increment of action potentials and membranes, and therefore it will be followed by single twitch, one contractions followed by one relaxations. And when the action potentials comes like the doubles to be two action potentials arrives, and it will triggers like two kind of physical activity of the neurons. It will induce one contractions, and it tend to be relaxed. But when the action potential arrive, it will induce the next contractions of the muscles, and therefore it's called summations. So summations, this will increase some. So the force, the strength of the muscle contraction will be increased by such a stimulations. And here when that stimulations come again with the frequent numbers, you see here that from one, and then these two stimulations, two action potential form, and this is four times of potential action potentials, it will triggers kind of this one contractions, slight relaxation, contractions, slight relaxations, and high concentration, high contraction force. The higher the stimulations and the higher the contraction force will be occurs. So here it is, they call it the tetanus stage, because why at this stage that this muscles could then relax perfectly, because every time it tries to relax, then the stimulations will induce its contractions, and it will increase the strength. The power for the contractions will rapidly increase. And here it is when the frequency, as you see here, like the frequency for action potentials related with the stimulations from the neurons increase in rapid way. Here it is that the muscle contractions, muscle will constantly contract without any particular relaxations. So here it's only contracts, and the power for the contractions will increase in such a height value, and afterward it will come to the state that there is no relaxation at all. So it is mean the tetanus, the perfect stage of the tetanus. So what is the tetanus? It's actually the constant contractions of the muscles without any relaxation stage. So here it is in the skeletal muscles. So when we compare with the cardiac muscles, the muscle composed of our cardiac, our heart, so it's quite unique because the cardiac muscles never experience such a tetanus like a skeletal muscles. So our questions is, here's our questions. What is the particular difference in electricity and action potential pattern between the cardiac muscles and skeletal muscles? And therefore it will we can explain that why then that cardiac muscles never experience such a tetanus stage. Could you imagine, for example, when our cardiac muscles undergoing this kind of the tetanus, that you will die in such a hard way, because for example the contractions of such a cardiac muscles less than 1 minute. Also the constant contractions without relaxation, it will stop the blood flow. It means that it may kill you in such a way. So for these questions, please, anybody can raise a hand for. Okay, Miftah, do you have any suggestions about the particular difference between the skeletal muscles and cardiac muscles based on its electricity?
Thank you, sir. I wanna try. I'm sorry if it is not correct, sir.
Okay.
I have watched your video before, and in your explanation about especially in refractory period, sir. So in this period, actually, skeletal muscle and cardiac muscle that have refractory period. So what is refractory period? Refractory period is there is tension, tegangan, and potential action potential that make this muscle always how to say it tidak akan pernah dia terpengaruh, the contraction is never terpengaruh because affected walaupun ada stimulus yang datang. So the differentiation between cardiac muscle and skeletal muscle is in the in action potential. So cardiac muscle that have a more longer action potential than a skeletal muscle, that's make cardiac muscle that's never seizure, kejang, seizure, because the refractory period that's absolute, that means never enough, continue. Like that, sir. Because of that, cardiac muscle that have absolute refractory period and skeletal muscle that hasn't. Maybe that my experience.
Okay. So the particular informations we can get from the Miftah explanations that it is related with the particular activity of action potentials, because the action potentials for the cardiac muscle is longer as compared with the skeletal muscles, and therefore because the action potential is the state where the muscles will never be affected by any kind of stimulations or other stimulation from the neurons or from the outside. It will never affect it, and it's mean that it will be resistance of the continuous stimulation that will end up with a tetanus. Anga, do you have any additional explanations or another variations of explanations related to these questions? Please, Anga.
Yes, sir. Maybe I add the answer from Miftah Ilmi. My answer is the difference is that striate muscle has very short refractory period, while the heart muscle has a long refractory period. And then we know that the refractory period is the muscle will not be affected by contraction activity, even though walaupun ada stimulus baru yang datang. Lalu pada otot lurik ini ketika ada perubahan potensial aksi pada membran ototnya, itu akan terjadi depolarisasi, maka otot akan mengiringnya dengan berkontraksi dan kembali lagi berrelaksasi. Dan pada otot lurik ini periode refraktorinya itu sangat singkat. Dan juga kalau misalkan pada otot jantung itu tuh berlangsung lama karena potensial atau pola aksinya itu berbeda. Karena di potensial aksinya itu terdapat delay pada atau perpanjangan waktu, sehingga durasi atau lama dia kembali ke posisi awal yang fase polar itu, maka pada membran otot maka akan berlangsung secara lama. Dan keuntungannya pada otot jantung ini, we know that periode refraktorinya itu kan panjang, sehingga berapa banyak pun stimulus yang diberikan selama periode ini, maka kontraksi otot tidak akan berpengaruh, dan itu sangat penting agar otot jantung itu tidak mengalami yang namanya tetanus atau kejang-kejang. Mungkin itu dari saya, Pak.
Okay, thank you, Anga, and also thank you, Miftah. So it is absolutely correct that the additional explanations also from Anga that as you can see here, the blue line is showing the electric activity. So it's called it action potentials or the electric activity of muscles. So here it is the polarizations and to the tops, and then will return back to repolarizations, and this is the basal stage or polar stage or resting stage. And when you see here that is really in short period of electric activity of this muscle, the skeletal muscles. When you look on the cardiac muscles, that here it is that of course it will immediately increase upon stimulations or upon impulse from the neurons, and then the membrane of the muscles will experience the increment of action potentials. But as you see here, as Anga state that there is a delay for when it phase will return back to the resting phase. So there is such a long period of this action potentials, and this is the particular concept that during this electrical action potential stage, these muscles will ignore any kind of stimulations. The next stimulations will be ignored as long as the electricity is still in this stage. So let's take a look at these comparisons between the cardiac muscles and skeletal muscles. So in skeletal muscles, I previously said that the length or the durations where the action potentials only happen in like 10 milliseconds, so the very short period, and it will be followed by the physical activity of the muscles, including contractions, and then relaxations. So 1% action potential in short term, and followed by the single twitch. And here it is for the cardiac muscles, like one stimulations happen here, of course it will trigger the similar pattern of action potentials increment, but there is a delay in such a long period. You see here that in skeletal muscles it will only take 10 milliseconds for completing this cycles from depolarization return back to repolar and polar. But here in cardiac muscle, it requires like 250 milliseconds. So it is mean this will be delayed like 50 minutes, 25 times slower as compared with the skeletal muscles. And the important is that whenever we apply the stimulations, or whenever the signal or the impulse comes right from the neurons to these muscles during these periods, it will ignore. So there is no contractions and relaxations affected. So this regular pattern will remain stay like that. And the next stimulation will be processed only like when the state of the electricity come to the normal or polar state. So only after 250, when the stimulations comes, then it will induce, of course, this depolarizations again, and will induce the next cycles of contractions and relaxations. So here it is, when you look at this skeletal muscles, when we apply when the stimulus come here and will induce the depolarizations in very short period, like when you apply the stimulations, or for example, when the neurons impulse from the neurons comes like in 15 milliseconds or even 11 or 12 milliseconds, it will induce the depolarization stage, and of course it will be followed by contractions and relaxation. So anytime when the impulse comes like in these black symbols, it will induce the contractions of the muscles, and eventually it will end up with the constant contractions, the sacral-tetanus. So here it is, you know, the greatness of the designs of our God to this particular muscles, that our cardiac muscle is protected from the tetanus, and we will never you know, experience such a hard situations when the cardiac muscles you know, in constant states like in contractions. So I think that's you know, for that sessions, and we come to the next questions. The last questions is about the energy or contractions of muscles. So for the contractions and relaxations, previously, all of you also have explained that actually ATP is the absolute requirement for contractions and relaxations of muscles. But can you define what kind of the energy resources for the muscle activity? And the next is, or how they use such contractions, and how they use such an energy resource. So let's start from, okay, I'm curious about Fauzia. So Fauzia.
Okay, sir. Thank you, sir. I will explain
Okay.
about some of energy in this muscle. Jadi sumber energi otot berasal dari tidak dapat dilakukan secara bersamaan, namun dilakukan secara berurutan, yang dimulai dengan penggunaan ATP yang siap pakai, kemudian ada metabolisme kreatin fosfat dan pemakaian energi dari glikolisis, dan serta untuk respirasi seluler yang terjadi di mitokondria. Jadi untuk ATP ini dihasilkan dari ATP yang sudah tersedia dalam pemecahan molekul yang kompleks dan menelan waktu yang cukup lama, serta untuk sumber ATP tersebut langsung dapat digunakan. Dan untuk ketersediaan adanya sumber ATP dari katabolisme kreatin fosfat yang proses pemecahannya lebih cepat. Dan terakhir itu juga berasal dari kondisi aerobik saat asam pirufat yang dikatabolisme melalui proses oksidasi aerobik yang menghasilkan ATP juga, dan hal ini akan merubah anaerobik itu menjadi asam laktat. Mungkin maybe that's all from me, sir.
Okay, Fauzia, so thank you. So next, Anjini, do you have an additional or the other versions of explanations how the muscles, what are the energy resources for the muscles and how the muscles will use it?
Okay, sir, I'll try to answer your question.
Okay.
But my question probably same with Fauzia. But I wanna addition a little bit to answer your question. And then my question are, the working of muscle will needs the big energy to open the process ion channels, and then to pump calcium ion to protect concentration gradient until change the conformation with how to say kepala-myosin when that occurs in contraction. Because of that, the source of the muscle is so important. And then there are some energy source for the muscle, but penggunaan ATP-nya itu will directly secara berurutan from the ATP that directly available until the result molecule is broken to the complex, and then there is needs the long time to do that. Dan terdapat sejumlah molekul ATP yang siap dipakai yang kemudian disimpan di dalam muscle cells until they are need to contraction, and then the ATP or ATP will use directly. I think that's all my questions, sir. Thank you.
Okay, thank you, Andy, for your additional explanations. That it's really important that you add information that ATP will be used first for generating the mobilizations of ions during the process of muscle contractions and relaxations, and also for the cross bridge between vitamin actins and the head of myosin. And next week, would you like Aryan, please? Aryan, do you have additional opinion or explanations about such a questions, whether about the energy resources for muscle contractions and relaxations? Yeah.
Okay, thanks, sir. I have the same explanation like friend Professors, but I will try to explain about process like creatine phosphate. First creatine it's will dibaca menjadi creatine phosphate and ADP. So dengan bantuan enzim creatine, it is creatinase, it will produce ATP. It is but dalam proses ini ATP yang dihasilkan it's like hanya bisa digunakan 15 detik. Lanjut ke proses selanjutnya, it is the glycolysis. Like as we know, glycolysis it is from asam pyruvate the ATP, but just 2 ATP in this produced in this glycolysis, but cause the ATP from glycolysis just cukup untuk 1 minute for muscle contraction. The next respiration, it is such as we know, respiration terjadi di mitokondria. It is like environment aerob, itu kayak membutuhkan oksigen. Seperti yang kita tahu, respiration in mitochondria it will produce banyak sekali ATP, like sampai 36 ATP dihasilkan. Namun like we know, cause the in respiration we need oxygen, cause sebabnya pada proses respiration itu tuh butuh oksigen dalam reaksinya. Terima kasih, sir.
Okay, so Aryan particularly explained about this process. So the first stage of energy utilization is coming from creatine phosphate. So CP is creatine phosphate, and it's mean the binding between creatinine and phosphates. Of course, when the enzymatic reaction triggers the phosphorylations, so the phosphate will be separated with creatinine, and therefore the phosphate from creatinine phosphate will bind with ADP and to be ATP, and therefore it will generate single molecules of ATP. And then the next process is glycolysis, using, of course, the glycolysis is the process of utilization of the glucose from the blood glucose or from the catabolism from glycogen, and it will produce also the ATP. And this glycolysis happen in cytoplasm of muscles. And afterward, the latest one is like oxidative phosphorylation in mitochondria as the next process of energy productions. And therefore, the amount of energy is like multiple levels increase, like 38 ATP produced by this process as compared with the previous, like in glycolysis only produce 2 molecules of ATP, and creatinine even only produce a single molecules of ATP per 1 molecule of creatinine phosphate. And it's mean that actually there are several energy resources for the muscle contraction. So the first is, of course, our muscles will use the ATP by labels in our muscles cells. So the ATP will be used directly. And when the ATP, but this is the fact that actually the free ATP molecules is only in like a very slow, very low concentrations in our muscle cells. And therefore, when our muscles only depend on such a energy resource from the available or ready to use of ATP, it will be limited. But afterward, there will be creatinine phosphate. So this creatinine phosphate is simple molecules, and to produce ATP, to produce energy from this process also requires only single step. So only like the phosphorylations by the enzymes and the separations of phosphate, part of this creatinine phosphate, and therefore it will produce ATP. But as previously mentioned by Aryan, said that this is only limited amount of energy process produced by this one, and therefore it requires the higher concentrations of energy, and therefore it will turn to glycolysis, so the utilization of glucose in cytoplasm through this process, and it will produce the ATP and also produce the pyruvic acid. And the last one, of course, it will the turn for the cellular respiration taking place in mitochondria. So call it that oxidative phosphorylations in this process, and it will use glucose and pyruvic acid and also fatty acid, and also this one the pyruvic acid also will be used in this one. And of course, based on these mechanisms, it will produce quite amounts of energy. But as you see here, this is the simplest one, and then it's quite complicated. When you learn about the glycolysis, there are several steps there, and it requires a lot of enzyme, a lot of steps, and it takes the time to produce ATP from this process. And then this is also the most complicated one, because it requires oxygen and any kind of substances for the process, and enzyme as well. So this means this is the largest choice for the muscles, not the first choice, because the first choice is the simple one to produce the ATP. Okay, so I think that's all. But when you focus in this one, in this drop, you can understand that actually when we when our muscle work in very short period, it only use the small molecules of ATP. So the free molecules of ATP, or ready to use ATP, ready to use forms of the ATP molecules will be used immediately. But because the amount of ATP in the free form is really, really limited, and it will turn to be the utilizations of creatinine phosphates, and therefore the red line is creatinine phosphate utilizations. So during the several seconds of the muscle activity, we use such phosphocreatinine systems. Phosphocreatinine molecules for generating the ATP, but the amount of ATP provided by such a process is limited, and therefore it will be glycolysis process, so the production for the energy. And also this is also has limitations because the glycolysis only like it is adequate for like 1 or 2 minutes of activity, but for long-term activity it should requires the aerobic phosphorylation, like something happen in mitochondria like this. So when you want to burn your belly fat or the utilizations or your fat and amino acids, we should do the long-term aerobic activity, like when you run, and probably you should run more than 15 minutes in order to reach the aerobic phosphorylation stage. But when you run like in a single minute, then of course it's only use the glycolysis. And when you just in slight movement, like in very light activity, so you only use the ATP from creatine phosphate, not from glycolysis, not from fatty acids. So the more active you use your muscles, like in intense activity for sport or any kind of activity, it will triggers the utilizations of various sources of energy, including amino acids and fatty acids, and also pyruvic acid as the product of glycolysis. Okay, so I think Aryan, do you have other options? Or
I have a sir.
Okay.
Jadi pertanyaannya, sorry in bahasa, sir.
Of course.
Jadi dari proses dari creatine fosfat, glycolysis sampai respirasi itu adalah suatu proses atau bagaimana, sir? Apakah ada di bagian tubuh lain yang dominasi oleh creatine fosfat proses atau glycolysis atau itu tuh? Thank you, sir.
So it's all happen in muscle cells. So in any part of our muscles, so even in biceps and triceps and in muscles in our leg, in any part. So this is taking place in cellular levels. So every single cells in every single muscle cells will use such a process. So phosphocreatinine also happen in the cells, in the muscle cells, and glycolysis also happen in that place, and oxidative phosphorylation happen in this place, at the same place, at the same muscle cells in every part in our body. But the priority of the muscle usage, there is a kind of sequential. So the muscle did not, the muscle cells don't use it in the same time. So there is a first priority. So the first priority is usually the free ATP molecules, because it's really easy to use it. You just only use it like when you want to buy something, you use a cash. You use cash money. That's kind of that's the simple analog for this one. So when the molecules of ATP just less or reduce because the numbers is really limited, so it will turn back to the second simpler one. What is it? The creatine phosphate. So it's happened in the similar cells in every part of our body. So there is no differentiation. Like for example, creatine phosphate happen in these muscles, and phosphorylated oxidations happen in this muscle. No, it's happened in every single cells of our body. But because ATP also available in the cells, in muscle cells, and creatine phosphate also available in such a plasms, and glucose also available in the muscle cells, and fatty acid, pyruvic acids, and amino acids also available in muscle cells. And therefore all the energy resources all used by the muscles, by all part of our muscles, but they will use it in sequential. So they will use the very simple way to produce ATP1, and when the numbers of such molecules and the numbers of ATP available for that, they will change, where they will shift to the next process. So from the simple process to be the complex process. So it is a quite logic actually. When you to buy, like when you want to buy food in cafeteria, you will use something that will be in practical way. You will use your cash money instead of using your mobile banking, for example, or you will use like ATMs or any kind of that. So the muscle also has the similar priority. They will choose to use the energy resource from the simple process the first, and then when they when the option is limited, then they will skip to the next, the complicated one. So that is Aryan.
Baik, sir.
Okay, so Miftah, do you have other questions or explanations?
Yes, sir. I have a question, sir. Actually, sorry, sir. When we back to muscle structure, sir, we can look. There is a different zone, right, sir? Like before we have talk more about in the zone, in the A band, or in the zone that myosin thick filaments meet with actin thin filaments. So there is complex mechanism in there. There is troponin, head myosin, and others, and there is how to say it, motor neuron axon and contractile filaments relationship. So my question is, what happened and what the occurs in the actin thin filaments without myosin first bridge, sir? Or in bahasa, apa yang terjadi di zona yang hanya ada filamen aktin tanpa ada myosin first bridge dan ya tanpa ada myosin thick filamennya, sir? What happened in there, sir?
Okay, first let's take a look in the structures. Probably it's better for us to see here. So this is actually the structural portions. So there is some zone that's like to make it easier to identify such a part actually. So there is a thick filaments and thin filaments in this region. So, but there is a part like a free of filaments. So there is no thin and thick filaments. So this like in this cross bridge regions, like the locations of attachment between actin and myosin. But in this free region, that's a part of sarcomere, Z-line. So there is no thick filament. So your question is about what happened in these regions. So actually this region is important for making that so kind of a constructive, this one. So this is the place for interaction between these thin filaments and thick filaments. So only in these regions. But what happened in this region? This region is needed actually for induction, because this is kind of let me show you this one probably. So as you see here, that actually the Z is the Z line, the filaments without the cross bridge region is needed, because this is the part that will strands and will restraints will become longer in these situations and will become shorter in this portion. So without this position, there will not be kind of the contractions of relaxation. So this in this regions you see here. So the green one is the location for the Z line. The free of there is no the activation, or I mean interaction between the actin and myosin. So that is important for these regions, because it's kind of the structural systems to make the muscles to be maximally contracts and relax at the some strains of that. So here it is, like you see in this one. This is also the region for the free of interaction between there is no interaction between actin and myosin in these regions in the Z line. But the Z line is important because this part is extended and shortened as the consequence of these interactions. That's a call it in sliding filament theory. Okay, so Miftah.
Oh yes, sir. I'm sorry before I out from the Zoom because the connection.
Okay.
But I got it. What you're explaining.
Okay.
Thank you, sir.
Okay, so that's part really important. We don't. That's part. So the the interaction between actin filament and myosin will have consequence in the contractions and relaxations. The contraction and relaxation requires such interactions, and also the motivation the other part, like in Z line. Okay, so everybody, since that we have a limited time, so now like 3 p.m. 5 minutes past 3 p.m. and I think it is the time for us to finish our lectures today, our discussion. Thank you very much for your active interactions, and I think we had such a nice discussion today talking about the muscle physiology, and actually there are a lot of process, a lot of mechanism that we need to discuss, but since that we just have very limited times and limited portions in this lectures, and I hope that you can explore for the next reference, and I hope also from our lectures, starting from the immune systems and endocrine systems and also nervous system, and today we finished talking about the physiological physiology of muscles. It also could gain your understanding on the process in your body self, and also could make you become wiser in facing the life situations, and probably I hope that you'll be successful. So in next next week you will take a lectures with Pak Robi, and it's mean that this is the last day we made in this lectures for this year in animal physiology lectures, and afterward we will probably meet again during the final examinations. So for the final examinations, I just inform you that we will have the oral examinations, and we also will have the writing examination. So there are 2 part examinations from me, and for the oral examinations probably through the Zoom. So I will call you one by one, and we will discuss about the similar discussion topic that we have talked in our lectures. So okay, so I think that's all, and if you have any further questions, don't be hesitant to contact me, to directly send me a message, and when I for example take a time to answer your question, be patient, because of course I don't know about everything. So I sometimes also consult with the expert, and also search from some credible reference, to ensure that my answer is supported by the evidence. And also, if you have any kind of idea, for example, you had idea for conducting research, for starting research in animal physiology field, so we are so welcome, because in my laboratory, in our laboratory, we also really welcome for the new students, for any kind of student, even you are starting from like first semesters of your study in university, we also so welcome. Please join with us. You can also watch some experiments, and if you have any idea, for example, you want to develop some drugs, you want to test some toxin from animals or from plants, so we are really welcome to collaborate with you, to discuss with you, and we also appreciate your idea for that. So don't be hesitant. You can visit our lab anytime, and you can also discuss with us to realize about your idea about research. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you so much, sir.
Okay, bye-bye.
Thank you, sir.
Bye, sir.
Okay.
Bye, sir.